sponsored by Directorate of International Program, IPB University, and PT Soho Global Health. This event, held by Tropical Biopharmaca Research Center, IPB University, from August 2nd to 10, 2021, with theme Herbal Medicine Utilization Biodiversity through Bioinformatics and Metabolomic Approach. Before we move to the next agenda, please let me introduce myself. My name is Sadila Vira Asoka as Master of Ceremony. I will accompany you through this event from the beginning to the end of the day. But I have information first, because today is Metabolomics Day, you have to install several softwares first. Link to download the softwares can be found on OCW website at section day 5. Once again, because today is Metabolomics Day, you have to install several softwares first. Link to download the softwares can be found on OCW website at section day 5. Okay, so without further ado, let's begin the agenda. First, we will have lecture from Dr. Sastia Pramaputri from Osaka University, Japan. She will present about what is metabolomics in food or jamu research theory part. And after that, maybe she will continue her lecture about metabolic fingerprinting for food jamu quality assessment. Now, she worked as assistant professor, Department of Biotechnology, Graduate School of Engineering, Osaka University, Japan, adjunct lecturer at Bandung Institute of Technology, Indonesia, and adjunct professor at IPB University, Indonesia. Okay, please listen carefully, make notes and questions, if any, to Dr. Sastia Pramaputri. Time is yours.
Hey, thank you very much, moderator, for the kind introduction. So do you hear my voice clearly?
Okay. Yes, we can hear you clearly.
Okay, so with that, I would like to start my lecture today. The first part is going to be on the theoretical part. So before I begin, I would like to greet everyone, my good friends from IPB. I see several names that are very familiar, Pak Rudi, Pak Wisnu, Pak Raffi. A very good morning also to all participants coming from everywhere. I see many international students as well as students from many different universities in Indonesia. I hope that today we will have a good and productive discussion after the lecture. So my title is actually about what is actually metabolomics and the importance of metabolomics in food or jamu research. I do several collaborative research work with Pak Raffi from IPB University on jamu research, but probably he will be talking more about that part. So I will be talking more on my talk about my experience to deal with food metabolomics. So because today is Metabolomics Day, I would like to remind you again why metabolomics is quite special, because it's one of the latest omics developed, and it's because the metabolome is the end result from gene expression. It's considered very closely correlated to the phenotype. And because of the many different diversities of metabolites present in a biological system, then we also have a long way to go to achieve the real metabolomics. I mean, the real metabolite plus omics. As we know that omics research always aim to do comprehensive profiling of the biological molecule. In the case of the genome, you would like to have comprehensive analysis of the genome. Transcriptome is the same for the RNAs. And for proteome, you want to analyze many different kinds of proteins. But for metabolomics, because of the very high diversity in the physical chemical properties of metabolites, in order for us to do a real comprehensive analysis involving very big, large coverage of target metabolites, we sometimes need to continuously develop the techniques to measure many different kinds of molecules having different chemical properties. We also have to develop a lot of library, internal library, in-house libraries, in order for us to do a good metabolite annotation. So later on, I would like to explain to you more about what metabolomics is and what kind of research is involved in metabolomics. So metabolomics itself is an interdisciplinary research where the knowledge of bioscience, analytical chemistry, and informatics is very important for a successful metabolomics study. Usually we start with a choice of biomaterials, and these biomaterials can be anything, because any biomaterials that used to be a living being would have metabolism capability. And since metabolites is either an intermediate or a product of metabolism, then any living biomaterials before would have many different kind of metabolites contained in it. We could choose the biomaterials ranging from microbe to human samples. And of course, depending on our study design, what we would like to extract, what kind of metabolites we would like to extract, we have to select appropriate solvents, chemical solvents for the extraction. And in this case, we need a good knowledge of organic chemistry. And then once we already have our extract, we will subject the extracts for analysis. And for this analysis, we also need a good analytical operation for analytical chemistry. So where we will use advanced instrumentation, most commonly used is mass spectrometry or NMR-based metabolomics. And then because it's omics by itself, naturally, we will have a generated data of big data. Then in order for us to visualize this data clearly, then we need a process called data mining. And we can do so by the operation of informatics. And for that, we need knowledge on statistics, of course, mathematics, and then we could visualize the data more clearly in order for us to understand the meaning of the data. So as I mentioned before, because the range, the diverse biomaterials of choice can be available for metabolomics research, the application area is also quite large. So I would just like to name a few of the examples that we do in our laboratory, in Fukusaki Laboratory in Japan. And it can be, for example, to understand the gene function and interaction in bacterial, plant, animal, or human research. It can also be for clinical research where we develop new diagnostic assays, or we could analyze or assess the effect of drugs to the outcome of a particular disease. And we could also have the application for bioindustry. This is one of the research topic that I have worked on for the past 10 years because I'm a trained microbiologist. So I use microbial cells as host cells to produce many different kind of compounds, useful compounds. Some of them are biofuels, or some of them are important biochemicals used in the bioindustry. And I work on dissecting these metabolic pathways inside these microorganisms in order for me to understand what kind of genetic strategies that we have to do in order for us to improve the strain and improve the production in the bioindustry setting. So this is more related to chemical engineering or biochemical engineering. And because our laboratory is under the Department of Biotechnology, we are in the Graduate School of Engineering. We do a lot of research on how we could optimize a system as engineers. And also, we have a lot of experience to apply metabolomics in food and agriculture. One of the ways we could apply this technology is by doing quality improvement and to assist breeders in strategic trait optimization, and also to improve the post-harvest management. So these are some of our research partners. In my research group, we divide mostly 3 different kinds of application area. One is more like medical and pharmaceutical, where we also collaborate, as I mentioned before, with Tropical Biopharmaca Research Center in IPB University for jamu research or herbal medicine, Indonesian traditional herbal medicine. and its relation to some health benefits in order for us to standardize the quality of the raw materials used for jamu preparation. We also have a collaboration with one of the renowned vaccine manufacturer from Indonesia. We call that Biopharma, where we together we develop the production of different biomolecules and assist them in the system optimization for production of these biomolecules. And then we also have a collaboration with Harvard University in the medical school for epidemiology-based research. And we would like to move to more like nutritional-based metabolomics, where we could investigate the effect of a particular dietary intake to health, to promote health benefits in humans. And we also have a collaboration with the University of Manchester for the production of antibiotics and how to optimize and how to understand the mechanism of how antibiotics production is produced, particularly in Streptomyces species. And as for energy, as I mentioned before, I have, this is actually the start of my career in metabolomics, where we work with UCLA, with NUS, and with RWTH Aachen, which is where Professor DJ Habibi, almarhum, used to study. We collaborated with these three universities, and they are all biochemical engineers, metabolic engineers, and we together, we assist them in the strain optimization and to increase the production of many different kind of molecules. With RWTH Aachen, for example, we collaborate to optimize the production of biopolymers. With NUS, we collaborate on optimizing the production of short chain branch fatty acids and very interesting like lipid-based compounds. With UCLA, we collaborate to improve the production of many different kinds of bioalcohols for energy production. And here down below, almost all of the research partners, many of them are actually different faculties and different research centers from IPV, where we collaborate in many different kind of research, starting from tempeh, seafood, tropical fruits, and also we collaborate with the Indonesian Coffee and Cocoa Research Institute for coffee and cacao research. We also collaborate with ITB professors on many different kind of research topics related to food, from fermented food, fruits, as well as shrimp. And the latest one we added into our collaboration just this year with two international companies. One is Novaton. This is a Swiss-based aquaculture company for shrimp production. And for Pipiltin, we collaborate on the fine chocolates from Indonesia. If you are from Indonesia, maybe you know this Pipiltin chocolate is one of our most famous fine chocolates that are sold in Indonesia. And the price is quite expensive, but they produce the chocolate in certain way that they manage to produce the chocolate from a single origin cacao beans. And later on, I will tell you more about all of these different range of application areas in metabolomics. So before we start, I would like to repeat again what I mentioned regarding about the workflow of metabolomics. Starting from sampling is very important. Then how you prepare the sample is very important. Then how you analyze the sample is also equally important. Because afterwards, if you do not do the sampling properly and you don't do the sample preparation and analysis properly, we say like it's junk in, junk out. So if the samples is already not incorrectly prepared, the data generated will also be incorrect. So then you will have an inaccurate data, and then probably you will not be able to generate a useful hypothesis or useful result from samples that are not prepared correctly. So in our laboratory, how we conduct the sample, how we prepare the sample, how we perform the sample analysis in very, very good controlled QC and the machine should be very well maintained is very important key of success in order for us to generate good quality, high quality data, trustable data in order for us to do our experiment. So when we would like to do experiments in metabolomics, the important point as a starting point is not directly to think about, okay, what kind of machine I should do, and oh, I don't have this kind of machine. No, but what's very important is to ask the right biological question. So this is the range of like the workflow of thinking in metabolomics research. First is you have to ask the right biological question, whether it's relevant or not to metabolomics research. If it's already a good, solid biological question, then you will go to the experimental design where you would decide the extraction solvent of choice, how to do sampling, how to optimize the sample preparation, and then the data acquisition, what kind of instrument you would like to choose for the target metabolites that you would like to analyze, the coverage of metabolites that you think would be important to answer your biological question. And then you do appropriate data pre-processing, and then you do the data analysis. Afterwards, then it comes the biological interpretation. So if you see from all of the cycle, it starts with a good biological question and it ends with a good biological interpretation. So people doing metabolomics without the knowledge of bioscience will have a lot of difficulties to make sense of the data. They will have very excellent generated analysis data, but then don't know what to do with it, don't know how to view this data into something that would be useful and will bring our understanding further in terms of the biological science or the bioengineering. So it's very important for us to consider how to make a good biological question. The first one is to start with a good working hypothesis. The second one, the analytical design, whether you want to go targeted into a particular target metabolite, or you want to do a non-targeted approach where you actually don't know what kind of classes of metabolites might be important to answer your question. And so you go non-targeted, you just analyze as many as you can. And then afterwards, once you understand what you would like to target or the coverage of your metabolite, then you decide the appropriate analytical instrument, and then the appropriate sample preparation method, appropriate way to validate your method, and then to do the data analysis method. Every single thing cannot just be a generic protocol. Of course, you start with a very generic protocol, but every sample is different. Even if we, for example, analyze mangosteen plant, if you analyze the fruit, you analyze the seed, or you analyze the leaf, or you analyze the stem, different parts even in the mangosteen plant will have to be treated differently as they contain different things, and the matrix, the biological matrices will be different. So it's very important for you to completely consider all of these things before you start a metabolomics research. Then I would like to bring your attention to the differences between targeted and non-targeted approach. As you know, there will be a trade-off. It's a give and take relationship between the metabolite coverage and the quality of metabolic analysis. If you aim to analyze non-targeted metabolomics, means it's like a metabolic fingerprinting. You analyze as many as you can and consider all of the features, then probably you have a low data quality. That means you have low quantification accuracy because you're measuring so many different things. You have probably low sensitivity because you have to cover so many things. And most importantly, you have low identity because it's not possible for you to do like metabolite annotation of over 1,000 metabolites at once. The best platform so far in the world that I know of is owned by a company based in United States. It's called Metabolon. They have their own internal library of 5,000 metabolites, and they use 4 or 5 different platforms to analyze all of the samples. And even that, they could get at most 500 annotated metabolites or less than 1,000. So if you would like to have identification of the metabolites, probably we will have a range of between 100 to 1,000. That's already very good. That's called metabolite profiling, where we have high complexity, multiple classes of metabolites analyzed at once. And if you would like to move to a more holistic approach, which means the real omics, we can go for metabolic fingerprinting with very complex profiles, but then it's maybe going to be quite challenging for the metabolite identification. And for example, if we just would like to analyze, like we want to analyze vitamins, and we would like to analyze only on the water-soluble vitamins, there's very limited compounds, and it's from one single class, then you could have a targeted analysis where you can focus on analysis of, for example, sugar analysis or amino acid analysis. These are multiple compounds, but within a single class, and then you could develop like one beautiful analytical method to measure this one single class. If your biological question can be answered, certainly, just by measuring this one single class. And if you would like to validate, for example, okay, from one single class, I know maybe among many different kind of metabolites that I analyze, I know amino acids are important. So I would like to just develop an amino acid analysis. And then from the amino acid analysis, I know that proline is important, for example. Then you could just have the single compound assay just to confirm the concentration of this particular metabolite of interest. And then you will have the highest metabolite data quality and accuracy. So to decide the appropriate analytical method, then this is like a diagram where you could measure many different types of metabolites. So usually we consider the size of molecular weight, like whether it's a small metabolite or large metabolites. Usually the primary metabolites tend to have a small molecular weight, whereas the secondary metabolites, it tends to have like larger skeleton of metabolites and therefore larger size. And we also consider the polarity of these metabolites. So this is a diagram where GCMS is very good for small hydrophilic metabolites mostly. And we could have in LCMS, we could have a wide range of metabolite analysis if we just change the column and so if we change the solvent in the column, then we could have a wider variety of metabolites that can be analyzed using LCMS. And these things are the consideration on where your target metabolites are. And sometimes it is also good to combine GC and LCMS all together in order for you to complete the metabolite coverage. Some of them probably cannot be detected by GC, some of them cannot be detected by LC. And so by combining the two instruments, you'll have a wider range of metabolite coverage. Okay, moving on for the sample preparation. I have already mentioned that the sample preparation may vary depending on the sample. It's quite different compared to, for example, how to deal with microbial samples, how to deal with plants, with animals, clinical samples, and food samples. Especially, you have to consider also if it has very high sugar, for example, or it's very oily, it's very sticky. The way to treat these samples might be different. So for the sample preparation, usually it's important to do the harvesting, which is the sample collection. And then this is a very important step in metabolomics to do after sampling is to do quenching. Quenching is the purpose of quenching is for stopping biological reactions in the cell. And it is very important to immediately stop metabolic activity, because what we would like to measure is the metabolite concentration at a particular time. This is if we do the snapshot metabolome analysis. It's basically like taking a picture of a particular portrait of the metabolite and metabolism in a given time. Then you need to make sure that the picture is taken in all different samples exactly in equal time. Otherwise, what you're comparing is not the same thing. So this is what I'm saying. Sometimes I see people comparing sample A, sample B, sample C, but they do not do the quenching uniformly. They do not do it in the same way, exactly the same way. So actually, for example, if we're talking about microbial cells, grows really fast, very fast doubling time. And you do this in 1 minute. This one, you let it grow for another 3 minutes. You let this one, you let it grow for another 10 minutes. By the time you do the quenching in the sample, actually you're already comparing microbial cells in different stages of growth. Then you cannot compare them in a similar manner. So it's very, very important for you to stop the biological reaction in the cell exactly at the same time and collect the samples that are exactly in the same stage. So this is the challenging part. It requires practice, it requires technique, it requires a very careful handling of the samples, especially if the samples are fast-growing samples in which very active metabolism still ongoing within the samples. And also, the next one is how to do the extraction. So the extraction is after you stop the metabolism, then afterwards you take the metabolites out of the cell. So for obtaining metabolites in the cell, we call it intracellular metabolites. We need to perform the extraction where we usually break down the cell wall or the cell membrane and then take out the samples. And we should do it in a very careful way so that when we do the extraction, before we take it out, we don't want the metabolites inside the cell to leak before we extract. It's called metabolite leakage. So the first step is to collect the sample and then quench and then extract. So the intracellular metabolites should be intact inside the cell. And then you break out the cell membrane, and then afterwards you take out the metabolites. But if by that time, before that, during the sample preparation, you're not careful, metabolites already leak because of improper handling, the cells already burst beforehand, then you already lose the intracellular metabolites. Then that will result in inaccuracy of the data, because what you're measuring is not the whole intracellular metabolite anymore, because some of them is already leaking out. So these things is very, very important for you to prepare the cells as a first step. Then for the data acquisition, there are many different kinds of analytical instruments that you can use. Usually the most common that we use in our laboratory is a combination between a separation technique and a detection technique. So the separation technique, we use chromatography techniques, either GC or LC. And before we also use supercritical fluid chromatography. We don't use that anymore, but we only mainly use GC and LC right now. And also we detect using mass spectrometer. Why mass spectrometry? Because it's very sensitive and it has a very good metabolite coverage, so it's suitable for metabolome analysis. So the principle of chromatography is basically just to separate the compounds. You do a separation before you detect it. So the compounds are separated based on the interaction within the stationary phase, and in this case, it's the column. So if the mobile phase is gas, the chromatography is called GC, and if it's liquid, it's called LC. And then this is the way that the flow of the mobile phase. If the compound interacts strongly with the stationary phase, it will be retained in the column and it is eluted late. If it interacts weakly, it will be eluted early. Based on the degree of interaction, whether it's strongly interact or interacts weakly, then the compounds will be separated through this chromatography method. So this is a very good way to imagine how metabolites pass through the column, and then they would have different interaction. So the interaction, for example, in the circle one is much stronger compared to the triangle one. And so the triangle one will enter the detector first, and the circle one will enter later. And we could see it in the screen as the elution. So the peak will be detected. For the triangle one, it will be eluted earlier, whereas in order, the circle one will be eluted later. So this is different kind of machines, and this chromatography technique is combined with MS. And the advantage of mass spectrometry is because the MS can detect minor component. We could get a lot of information of components, and it can be connected to different kind of chromatography. The weak point that I would probably not say in detail today, but it's very bad quantitative quantitativity, because the ion suppression is an unavoidable problem in mass spectrometry. So you actually cannot get like absolute quantification just by doing MS. You need to develop additional method like isotope dilution method, a particular additional method in order for you to get a quantitative data. So what you can get from metabolome analysis using mass spectrometry is mostly just relative concentration. It's not absolute concentration. And then it doesn't have really good reproducibility because we have a lot of variation. So if you do the analysis today and then you do the same analysis next week, the data is not exactly the same. So what we are checking is usually just the trend is the same. So if you're comparing sample A, B, and C, and then you see, for example, sample A has high concentration of amino acids, sample B has high concentration of glucose, for example, we just would like to see that the trend is reproducible, but the actual value cannot be the same each time. So that's why we call it relative concentration. You just compare things relative to each other. And also it's expensive. So this is the detection principle in mass spectrometry. Compounds in the samples is firstly ionized, and then it's separated according to the M/Z, the weight of the molecules, and then it's detected as an ion intensity. So first, the sample is introduced, and then there's an ionization unit, and then there's a mass separation unit, and then afterwards it's detected. So in chromatography, you remember that you have like a chromatographic profile where you could see the compounds are eluted depending on the retention time. Retention is the interaction between the sample and the stationary phase. And then afterwards, for the mass spectrometry, you could see that they are they will be ionized, and then you could detect the total ion intensity, and then you could check the M/C mass fragmentation pattern of each of these metabolites. And by the combination of chromatography, the retention time, and also the mass pattern, the mass fragmentation pattern, then you could do the metabolite annotation. Then you can know the identity of the sample. So this is just an overview of GC. First, you do the sample preparation, and then the sample is injected, and we use the gas as the mobile phase, and then the separation. Sometimes we use hydrogen, sometimes we use helium. Many different kind of gas can be used. And then afterwards, we use it in the column. The samples move inside the column, and then afterwards it goes to the detector, and then the first step is the detection. So this is how the GCMS data looks like. So GC or LCMS data, we have like 3-dimensional data. So first, the first one is the chromatographic profile. So you could see the elution. The first one is the retention time, or minutes in minutes. This is the chromatographic profile. And then you get the ion intensity. For example, the concentration of the metabolite is determined by the ion intensity. And then you have also the M/Z here. Then you could check from each of the peak that you measure in the total ion current chromatograph, the TICC, then you could see that different metabolites have different mass spectra. And from here, you could see that each metabolite will have a unique mass spectra, and they will be eluted differently based on its polarity. Then afterwards, you could do the metabolite annotation this way. So when you have, this is like an example of a chromatograph, you have many peaks, because as I mentioned, what we're trying to do is omics, metabolomics. Then we try to analyze the crude extract containing many different kinds of metabolites in a single sample. So then what we have will have many peaks at once. But these peaks are not always real peaks. Sometimes it's just instrumental noise, the small ones. So how do we determine which one is noise, which one is peak? And maybe some samples are some metabolites are eluted together, the co-elutes. Then afterwards, we need to process the data first in order for us to check and remove the instrumental noise by data processing. So that in the data pre-processing, we have to remove the noise and to determine which one is peak. There's a way to do that, the baseline correction, and then the peak detection. And then we have to align the same peaks are aligned in the same way in all of the samples. And then we have to do the deconvolution, for example, to remove, to make sure that the compounds that are co-eluted can be discriminated, and then do the normalization. And afterwards, we could result something like a data matrix like this, a generation of the data matrix. Then we have the data pre-treatment, where we could treat the data in many different ways for the scaling. And then we could go for multivariate data analysis. I think some of you who are here today probably are quite familiar already with this process. And here is the example on how we do the peak detection. So we need to determine which one is the significant peaks from the chromatogram. Sometimes the background is quite high, and these small peaks are considered noise, and these are considered peaks. So based on some criteria, we could say usually what we see is the signal to noise ratio. We could say, for example, signal to noise ratio has to be higher than 5 in order for it to be considered as a peak, or signal to noise ratio should be 10 for it to be considered as a peak. You could determine this for this peak detection, and the shape of the peak and the peak height value, or the area value as well. We could set up this criteria. And for the height value, it's just considering the height, it's the intensity of the peak. But the repeatability depends on the shape of the spectrum. As for the area value, repeatability is always good, but it doesn't depend on the shape of the spectrum. So area value is usually used compared to height value. And this is what we call in the S to N ratio. So the limit of detection, if we consider that it's just to detect, not to quantify, we could use like S to N ratio equals to 3. And for the limit of quantification, S to N ratio equals to 10. So we have a higher S to N ratio when we would like to quantify a metabolite. But if we just would like to detect it, we could set up the number to be lower. So this is what I want to talk about coilution, because as I mentioned, we are analyzing raw extract. So crude extract means that so many metabolites might be just coeluted exactly at the same time, because they have almost similar time when they are eluted. So you could see here, the shape of the peak is not as beautiful as this one. There's like a valley in here. And usually this kind of shape of peak in the chromatogram indicates that there are more than 2 metabolites, at least 2 metabolites in that peak. And here it can be actually 4. So this kind of peak actually in truth is something like this. Then the process of separating here into actually 3 different peaks is called the deconvolution. And luckily now almost all of the instrumentation have the feature of deconvolution. So the software is quite smart already. Now we could have this deconvolution software integrated into the analysis, and we could do the deconvolution quite easily using the software provided by the instrument company. So this is about the data mining. This is how the GCMS chromatogram look like, and then after it is being pre-treated, we have like the chromatogram looking like the one below. And then we do the peak identification, peak list, and then the multivariate analysis. Okay, so for the principal component analysis, this is the most commonly used unsupervised analysis, where the PCA rearranges the data into principal components such that the variance between samples are maximized. So we would like to see what would be the greatest factor that separates our sample non-biasedly. So this one is by looking at what kind of samples have the largest variance with each other. That's how we discriminate or differentiating samples. So here we have the sample and we have the retention time, we have the M over Z, and then we would like to take multiple factors at once because all of these samples have different retention time of the metabolites, different M over Z, and different total ion intensity. And then we could do what we could do from this really complex data. The PCA can just maximize the variance between samples so that here it will be fixed into the PC1 so that the data difference becomes the biggest and the data variance is biggest along principal component 1. And here we could have what we call a score plot. This is a plot that is made when PC1 and PC2 are drawn as the new axis. The score plot will look like something like this, where the PC1 is the greatest proportion of variance between the samples that lie along the PC1. In short, this is the score plot where the samples, each of the dot is one sample, one replicate. And then if they are clustered very far along PC1, which is usually the X1, then it shows that these samples have the largest variance in terms of their metabolite profiles. Then the PC2 is the subsequent PC, contains the next biggest proportion of variance between samples. So other than score plot, we also have loading plot. So in order for us to put the information into the PCA, usually we have to put the information in the metabolite data, what metabolite we annotate and their intensity. And the loading plot shows how much variable. And in this case, the variable we use is the metabolite data. So how much of each metabolite contributes to each principal component. So here before in the score plot, we see that each dot is our sample. And in the loading plot, each dot is our metabolite data. Then it will show what kind of metabolite is making sample A and sample B very different. Then you could see what are the metabolites that give the highest contribution to each principal component. In short, it will tell us what kind of metabolites are very different among different clusters of samples observed in score plot. So this is an example of the score plot using a sample of herbal medicine. This is Chinese herbal medicine that comes from different area, and also it has different quality. So for example, like samples from this one, Hokai Toki, Yamato Toki, and the high-grade Toki are clustered differently. And they found that, for example, samples from Hokai Toki, from Hokkaido, have higher concentration of terpinin and beta-myrcene. Whereas the samples from Yamato Toki along PC2 are separated with the high-grade Toki or Angelica acutiloba, and the metabolites that are responsible for this separation is expressed in the loading plot. So until here, this is just some of the theoretical explanation on how metabolomics can be done and what are the steps that are important to consider when doing metabolomics research. So this is the first part of my talk, where the conclusion is metabolomics offers an interdisciplinary approach for diverse research area. And we have a choice of a wide variety of biological samples, and we can use many different kind of instruments and techniques, and the most common being mass spectrometry based chromatographic techniques. And it could give a strong pipeline for metabolite identification and data mining. So metabolite data identification and metabolite identification and data mining are both crucial for the success of metabolomics research. So in 2014, Fukusaki Sensei and I summarized all of our lesson learned and summary of our protocols in our laboratory into this book. It's published in 2014 by CRC Press. It's available in Amazon. And the title is Mass Spectrometry-Based Metabolomics, a practical guide, where we lay out each of the steps that I described above, and we make a step-by-step protocol as well for many different kind of samples, and how to do the GCMS analysis, how to do the metabolite data annotation and everything. So it features a very simple, easy to follow, user-friendly style, and summarizes all steps in metabolomics research, from experimental design, sample preparation, analytical procedures to data analysis, with some illustrations as well. And as I mentioned, we also feature the optimized protocols and include a link to our in-house library. So if you use the same machines and you follow this, then you could have the access of using our in-house library as a tool to help you annotate your data. So that is the first part of my talk. I think I would end it here and have some discussion before we move on to the second part of my talk. So with that, I would like to thank you for your kind attention and return my talk back to the moderator. Thank you.
Okay, thank you so much, Dr. Sastia Putri. It's such an interesting topic. As Dr. Sastia said, before we move to the next lecture, is there any question from students? You can ask Dr. Sastia directly by raising your hand and unmuting your microphone. Please, I invite you. Is there any question? You can ask about the procedure, the theory, or the application of metabolomics, maybe. Okay, maybe I want to ask a general question. Some participants may be still curious why metabolomics approach is highly demanded on this day. Is it because the instrument also develops or something, ma'am?
I think in the past, because metabolomics is relatively new and the cost to do metabolome analysis was really expensive before, but now it's becoming more and more common. So the cost to do the analysis per sample is also decreased. That is one. And the second one, also, there are many more reports on the utility of metabolomics in many different fields. So people coming from different disciplines that are not familiar with metabolomics before also start to become interested. So in the beginning, maybe mostly people working on clinical research are doing metabolomics in the first stage of metabolomics development, but then the application area widens. And so it attracts people from many more various disciplines wanting to do metabolome analysis. And nowadays there are so many one package, tailored made one package offered by vendors that makes it very easy to use. Before, there's so many difficulty in the sample preparation, the know-how, we have to optimize it one by one. So only specialists can do it. But nowadays, the softwares available, for example, like I mentioned, the Deconvolution software, before we have to do it manually. And the metabolite annotation, we have to do it manually. But now with all of the development in bioinformatics and the advanced instrumentation also makes it much more easier and user-friendly. Many people who are non-specialists can use it also. So I think that expands the application area of metabolomics even more. I hope it answers your question.
Yes, it's answering the question. Okay, we have a question from Raden Bayu from Universitas Pajajaran. He said, thank you for your lecture, Dr. Sastia. I have a question. Is there any certain sample conditions that force us to only use certain instrument analysis? For example, NMR only or LCMS only, or we can generally use any instrument for any samples? That's the question.
Thank you. It's an excellent question. Actually, the NMR and MS based are very different in one, sensitivity, And second one is in whether it's destructive or non-destructive. So for NMR, you could analyze your samples, and then you could get your samples back. It's non-destructive, so that is a good point about NMR. But it's also much less sensitive than LCMS or MS. So in that case, you need to have large amounts of your sample. If you have small amounts, you do NMR because then you cannot detect anything because of the lower sensitivity. But if you have a very precious sample, but in larger amount, the good point about NMR is that you could retrieve your sample back and use it for other analysis. Whereas for MS, as I mentioned, you need to do the extraction. It's also very laborious. NMR is very easy. You don't need to do the extraction, and it's non-destructive, so you could just put it in the samples after very minimum sample preparation. So it's relatively easier to operate. Whereas in LCMS, you need to do like the extraction, sometimes derivatization, and then after you already do the extraction, you cannot get the sample back, it's gone. So, but if you would like to analyze many different kind of samples, sorry, not many different kinds of, if you would like to analyze many different kind of compounds, and the compounds are present in your sample in small concentration, then LCMS would be the appropriate method to use. So it's not just you can use any instrument for any sample. It depends on your sample. Because of the two issues, I think mostly the easiness of operation, the sensitivity, whether you have large amount of your samples or not, and whether you really need the samples back. For example, some samples are very, very precious, taken from, let's say, COVID patients, and you cannot afford to retake the samples again, and you would not like to use these samples because after the analysis you cannot get it back. NMR would be an appropriate method of use. So yeah, in short, it depends on your sample. I hope that answers your question.
Okay, thank you so much, Dr. Sastia, to your answer. There are questions from Faisal Maulana from IPB University. Actually, there are two main questions. You can answer it one by one. The first question is about the details. What research that Osaka University and Biopharma collaborate? Is there any journal published with Biopharma? That's the first question.
Okay, so it's relatively new collaboration. Actually, we made the collaboration just before COVID-19. So it's 2019, and we have some interesting results. The collaboration is on how to optimize the medium used for biosimilar production. So Biopharma is developing the production of several biosimilar molecules that can be used for anti-cancer therapy, and they would like to produce it in a cheap and efficient way. And we try to optimize by looking at the medium of growth for the cells that they use as a host for the production of the biosimilar that they are working on. And we try to check what kind of medium component that we could optimize further in order to increase the production of their biosimilar. In another application where we could do that is also by optimizing the process of the production in order for us to have a more optimized production process. Unfortunately, this research is now on hold because to 2020, early 2020, where we're supposed to do additional experiments on it, COVID-19 happened, and Biopharma is now extremely busy to switch all of their focus now on handling the pandemic and research on vaccines, especially on COVID-19 vaccines. So we have, I think the manuscript is 80–90% finished. So we would like to wait a while. Once we could complete the remaining 10%, then we could publish the paper, or we could probably just go ahead and try to publish as it is. So there's no journal yet, but we have already prepared the manuscript and it's nearly completed. And the second question, in our laboratory, the research that focuses on metabolomics is supported. Active. Okay, in treatment of diseases. It's not my own research project. It's Professor Fukusaki collaborates with medical doctors in hospitals to treat many different kinds of diseases. Not only medical doctors, but also dentists for periodontal disease, for example, and to find the active and inactive compound in the disease treatment. Yeah, he did that kind of research, but it's very, very minimum. Sometimes we are never the PI of that kind of research, because such clinical based research has to be led by principal investigator, the leader has to be a medical doctor. So we are usually just are the collaborator to help them analyze their samples, and then they're the ones who's going to lead the direction of the research. So for myself, my research topic, because I'm a microbiologist and I have some knowledge on food and agriculture because I'm a biologist, so I would like to stick to my own expertise, something that I'm actually familiar with. So yeah, I don't handle a lot of research on the diseases or something that is very much related to clinical research.
Okay, thank you so much for the answers, Doctor. Turns out there is one more question from Patrius Atma Jogja. How many sample is needed to make metabolomic one species plant? Is that clear?
I'm sorry, I couldn't catch the answer. Could you help to rephrase the question? Do you mean that how many samples is needed to do the metabolome analysis for one plant species? Is it the question, perhaps?
Yeah, maybe that's what he or she means.
The number of samples we have to determine based on the experimental design, based on the biological question, and then from that on, we have to determine what samples we would like to compare and how many. But as a rule of thumb, for one sample minimum, we need to have triplicates for one sample, one variation. So one triplicate, and then if it's a time course sample, we need to take, like, for example, you're comparing one species, but maybe you would like to monitor the growth, or you would like to monitor fruit ripening. In that case, you need to have all of the samples that represent the ripening stages of that fruit. Then from each of the samples, you need to have at least triplicates. So the sample, we usually try to limit the sample, not too many, 50 samples. But in the case of cohort, for example, people would like to check the effect of a particular drug in the outcome of disease, or particular dietary intake in a cohort study. So it involves a very large population. It can be more than 1,000 samples for one cohort, even sometimes 10,000 samples. So it really, really depends on the study design and the experiment that you would like to do for the sample.
Okay, thank you. Actually, there is one question. Is it okay to answer it? Oh, okay.
Yes.
There is question from Vinda Maharani from UNS. Unisba. Thank you for your amazing material, Dr. Sastia. I have a question about, is there a technical differences like extraction process or identifying processes for targeted and untargeted compounds?
Yes, it's very different. Actually, if you would like to target, for example, one class of compound that you know it has a small and then it has high polarity, then you can use a polar solvent to extract this solvent. But if you would like to do the untargeted compounds, means that you have to try to extract as many as possible. In that case, you need to have a solvent or solvent mixture that can extract compounds having different polarity, having different physicochemical characteristics. So you need to have a combination of different kinds of extraction solvent. And sometimes for untargeted metabolomics, there's also a technique where you don't do the one extraction, but you do 2 times extraction. For example, like one sample, you have the hydrophilic fraction and you have the hydrophobic fraction, and you process it differently to target, for example, the lipids and the non-polar metabolites. And when you want to target the polar metabolites, or if you want to target volatiles, the compound that can evaporate, the volatiles, then you also have to have another technique for extracting these volatiles that is different with the non-volatile. So there are many choices on how to do the extraction depending on the target metabolites that you would like to target. And you need to optimize this for different kind of samples. So in our laboratory, maybe this process to optimize takes about 6 months. Each student have to do their own optimization in order for them to have a good reproducibility, they have a good target, they have a good metabolite coverage, and they have a good accuracy in their analysis. So we have to consider many different things in order for us to have a good quality metabolome analysis. So usually it takes about 3 to 6 months to optimize this.
Okay, thank you, Doctor. Maybe one more question before we move to the next lecture. Yeah,
okay.
This is a question from Susilo from
Okay.
Universitas Muhammadiyah, Professor Dr. Hamka. I have a
Okay.
question. What is the difference between metabolomics research for master's and doctoral levels? That's the question.
Okay, this is very interesting question, Prof. Hamka. I'm actually a junior lecturer, so I'm not sure if I can answer your question satisfactorily. But in my case, I discriminate 3 different levels of research. For example, for bachelor research, I just give them a topic that we already consider and propose, and that kind of topic will definitely be finished within 1 year. And usually it's just a repetition of techniques. So the most important thing for the bachelor is to learn the techniques, and they are able to repeat it. And then they will be given a simple biological question in which they could answer satisfactorily within 1 year. For masters in metabolomics, usually they already have to know how to do the metabolome analysis, and they have to propose their own research proposal, their research idea, and they are demanded to have a polished analytical thinking where they could generate the new hypothesis based on the data generated from their metabolome data. But we do not usually within the 2 years, the metabolomics research in master course can achieve, for example, clue. We could get a very good clue as to what kind of metabolites is important to discriminate this sample and this sample. They could check like the variety or the diversity of different kind of cultivars or origin, and then indicate what kind of metabolites are different, or they could suggest potential metabolites that might be correlated with a particular food quality. But then for doctoral levels, novelty is very important. It has to have breakthrough and novelty, and we have a demand for them to publish 1 or 2 international publications to graduate from PhD in a peer-reviewed journal, international journal. And the study has to be correlated to one another to make a comprehensive and continuous research. So in Osaka University, actually, master and doctoral level is very, very different in terms of what we demand from them. For doctoral levels, they have to be able to demonstrate new ways, new method, or they should be able to bring a new understanding of a particular biological phenomena or they could come up with a significant improvement in a biological system. So not only to indicate this is a correlation between this and this, but they have to prove that correlation and then improve it further. So they have to do something about the metabolome data. The metabolome data is just the first step. And then what they do with the metabolome data to extend the knowledge of something else, and it's validated and improve it and create something new, is what makes the biggest difference between the master and doctoral level, at least in my laboratory. That's what I expect them to do. It's quite challenging, but they're doing really well, mostly. I hope that answers your question, Prof.
Okay, thank you for the answer. Maybe we move to the next lecture. To those who have questions still, don't worry. Dr. Sastia will continue her lecture about metabolic fingerprinting for food or JAMA quality assessment. Still, you can write down your question on the chat column